All right, we're continuing with glycolysis, and what we're looking at is what happens to one molecule of glucose as it goes through the 10 steps of glycolysis. In steps 1 through 3, glucose has been phosphorylated. It's been given 2 phosphates. Those phosphates came from ATP. And now we have something called fructose 1,6-bisphosphate. That's where we are. We have a 6-carbon molecule with 2 phosphates attached. We haven't made any ATP. We haven't seen any redox reactions yet, but we're going to see what happens in step 4. Okay, in step 4, this 6-carbon sugar is split into 2 3-carbon sugars. Guess what? This is the sugar splitting step of glycolysis or glycolysis, the splitting sugar step. So there's an enzyme called aldolase that splits our 6-carbon fructose into 2 3-carbon molecules. One of them is called dihydroxyacetone phosphate. Dihydroxyacetone phosphate. I'm just going to call it DHAP. We're not super interested in that guy. We're more interested in this guy up here, glyceraldehyde 3-phosphate. By the way, I think we're on page 13 in our PowerPoint slides. Glyceraldehyde 3-phosphate is how I'm going to write it. This is a molecule you should recognize and you should know about. Glyceraldehyde 3-phosphate, actually I'm going to write that up here. G3P, glyceraldehyde 3-phosphate. It has 3 carbons. On its 3rd carbon, it has that phosphate. It also has a carbon-carbon double bond there and a hydroxyl group there. So here's our molecule, 3-carbon sugar with a phosphate attached. Just like this one, but a different arrangement. Now, what's exciting about this step is we've split the sugar in half, and we're going to use one of these molecules in future steps, in steps 6 through 10. We're going to use glyceraldehyde 3-phosphate in steps 6 through 10. Wait, what happened to step 5? Well, in step 5, it turns out that the 2 molecules, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, can be interconverted. Okay. Dihydroxyacetone phosphate can be converted into glyceraldehyde 3-phosphate, and glyceraldehyde 3-phosphate can be converted into DHAP with the use of an enzyme called isomerase. But that's step 5. But what happens in glycolysis is glyceraldehyde 3-phosphate gets used in the next step, in step 6. And so DHAP gets converted into glyceraldehyde 3-phosphate. And then it's used up in step 6. And more DHAP gets converted into glyceraldehyde 3-phosphate. And it gets used up in step 6. And more DHAP gets converted into glyceraldehyde 3-phosphate. And it gets used up in step 6. So what essentially has happened in steps 4 and 5 is we've taken this 6-carbon sugar, cut it in half, and essentially made 2 molecules of glyceraldehyde 3-phosphate that we're going to follow through steps 6 through 10. So we're really going to have steps 6 through 10 happening twice because we have 2 molecules of glyceraldehyde 3-phosphate at the end of step 5. And that's how you can think about it. Okay, so we haven't made any ATP yet. We haven't done any redox reactions, but step 6 is super important. All right, and so you're going to look at slides 14 and 15 here as well as 13. In step 6, glyceraldehyde 3-phosphate gets converted into another 3-carbon molecule called 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate. We're rearranging this molecule, and this molecule is no longer a sugar. Something happened to it. Well, we have to look at what changed. When I look at the molecules, really the only thing that changes is what's going on in carbon 1 of this molecule, right? Up here we've got a hydrogen attached to the carbon, and over here we end up with a phosphate attached. Like this hydrogen gets removed somehow and a phosphate gets attached. Okay, this is a really important step. This, oh wait, we haven't finished it yet. We have to add in the other reactants. So glyceraldehyde 3-phosphate is a reactant, and so is this thing called NAD+. Nicotinamide adenine dinucleotide. Just call it NAD+. NAD+ gets converted into NADH plus an extra proton and another reactant here is what's called an inorganic phosphate, a phosphate molecule just hanging out in the cytoplasm, okay? Whew. So we've got lots of reactants. We've got glyceraldehyde 3-phosphate, NAD+, and we've got this phosphate. What is going on here? All right, this is a redox reaction. Let's write a few things down. This is a redox reaction. It's our first one. By redox reaction, remember, in a redox reaction, one of the reactants loses electrons and one of them gains electrons. And in this process of glycolysis, we expect glucose, or what's left of glucose, to be oxidized, to lose electrons. And that's what happens in this step. When this hydrogen, actually a proton and 2 electrons, are removed from this molecule is oxidized. It loses electrons associated with that hydrogen. And NAD+ is the reactant that picks up or gains those electrons. Okay, so there's a transfer of electrons from glyceraldehyde 3-phosphate to NAD+. They're actually it's a fall of electrons to a more stable location. Okay, so it's a redox reaction and some electrons are removed and transferred to this thing. What is NAD+? Well, page 15 shows you a picture of this molecule. It's huge, but, and I don't need to show it to you. You can take a look at it. But what's important about NAD+ is a coenzyme. And what's interesting about this coenzyme is it can be reduced. It